We are writing to you on behalf of a number of scientists to communicate a matter of deep concern. So began a short letter published in the leading journal Science in September 1973. It continues with a short technical description of the recently reported research that so alarmed the authors and others at the conference in New Hampshire that the letter arose from. We presently have the technical ability to join together DNA molecules from diverse sources. Scientific developments over the past 2 years make it both reasonable and convenient to generate overlapping sequence homologies at the termini of different DNA molecules. The sequence homologies can then be used to combine the molecules by Watson-Crick hydrogen bonding. Listeners of this podcast may be able to follow what they're saying, but if not, the next section of the letter is a little easier and it encapsulates the core concern, one that I think continues to resonate to this day and has had wide-ranging implications for biosafety policy used in laboratories around the world. This technique could be used, for example, to combine DNA from animal viruses with bacterial DNA or DNAs of different viral origin might be so joined. In this way, new kinds of hybrid plasmids or viruses with biological activity of unpredictable nature may eventually be created. The letter was authored by Maxine Singer, a Washington, D.C.-based molecular biologist who was just as passionate about how science is done as about the science itself. She was one of the co-chairs of the 1973 conference in New Hampshire that inspired the letter. It was followed by another letter published in the journal Science in 1974, written by Paul Berg and a number of other scientists who were developing these scary new technologies, volunteering to put all this work on hold and calling on all other scientists around the world to do the same. Given that very few scientists around the world were doing this sort of work, the intended audience was pretty small, and they did indeed largely heed this call. All this tinkering of DNA was put on hold till, as the letter says, an international meeting of involved scientists from all over the world should be convened early in the coming year to review scientific progress in this area and to further discuss appropriate ways to deal with the potential biohazards of recombinant DNA molecules. Maxine Singer stepped back in at this point to help organize the international meeting, bringing together not just scientists but also journalists, lawyers, and philosophers to define policies for DNA technology going forward. The conference was held in 1975 in Asilomar, a couple of hours south of San Francisco on the California coast. The geography matters, as you'll see all the early history of DNA technology revolves around the San Francisco Bay Area, which remains to this day a global hub of biotechnology. The 1975 Asilomar Conference is often pinpointed as the pivotal point in regulating recombinant DNA, and it did set the basic approach still followed today, that as far as possible, any DNA molecules made in a lab should be treated as fundamentally different from naturally evolved molecules, and they should be handled as if they could be dangerous. But the conference itself only made recommendations. Maxine Singer took those recommendations and made sure they got turned into a set of defined rules by the National Institutes of Health and anybody receiving funding from them. These rules include things like treating all lab-made DNA as biohazardous and disposing of it in the same way as you would live bacteria or medical waste. Since almost all universities receive NIH funding, these guidelines apply to most research labs in the US and have been closely mirrored in binding policies that apply in research systems in other countries. So while she by no means was the only person involved in the story of biosafety and recombinant DNA, she has probably had the greatest single influence. If lab-made DNA is treated as a health hazard in the labs themselves, then it doesn't seem to make sense that much of the globe willfully eats it every single day in the form of GMO crops. And that seeming contradiction has been crucial to the opposition against GMO crops from their first day. And yet ask many scientists, and they'll find the idea of being afraid of engineered DNA to be silly and illogical. And yet, remember, it was scientists themselves who first sounded the alarm. To understand why, you need to know a little about the technical details. What actually is recombinant DNA technology, or as we call it more often, genetic engineering? Welcome to Modified. I'm your host, Dr. Orlando DeLange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 9, The Early Days of Genetic Engineering. In this episode, you'll learn more about the process of creating genetically modified organisms, starting with the OG GMOs, genetically engineered bacteria. We will dive into the work that was happening in labs in the 1970s to cause so much concern, and find out how it fundamentally changed ideas about what could be done with DNA, triggering an immediate explosion of applied research reminiscent of the immediate application of Mendelian genetics to agriculture after its mainstream embrace by scientists in the early 20th century. And indeed, this new DNA technology was immediately applied to agriculture, giving us the GMO crops we know today. However, it took a little more time, basically because plants are slower to grow and harder to work with than bacteria. So for this episode, we'll focus in on the bacteria and get to know some of the techniques and terminology of genetic engineering, which we'll pick up in episode 10 to see how this was applied to crops, applied to plants to create genetically modified crops. As is so often the case in science, the key pieces of the technology needed to engineer genes were discovered during basic research, just trying to understand how DNA works, specifically how DNA is exchanged between bacteria. Bacteria are all around us. They make up about 12% of total biological matter on our planet, despite each bacterial cell itself weighing only about 1 picogram. That's 1,000th of a billionth of a gram. Right now, there are trillions of bacterial cells inside you, living in your gut, on your skin, and all around the openings and exits to your body. They are on every surface in our homes, and if we step outside, they are a ubiquitous feature of natural environments. You mostly cannot see them, but they are coating every leaf, embedded in the soil, and swimming freely in every body of water. Bacteria diverged from us in the tree of life about 3 billion years ago, and all that time they never felt the need to change the major body plan from being small, single cells, each one equipped with a relatively small set of genes that give it the information to produce a set of proteins that allow it to harvest material from its environment and to reproduce. As they grow, they eventually reach a point where the DNA inside the cell is replicated, and then the cell itself divides in two. All the resources of the cell are divided, and what was once one cell becomes two smaller, identical daughter cells. This is the process of binary fission that has served bacteria well for billions of years. But as scientists studied the genetics of bacteria in the early 20th century, several groups around the world noticed that genetic information didn't seem to pass vertically down generations as expected, at least not all the time. Instead, it seemed that genetic information in bacteria can be infectious. Introduce some cells that have some particular traits, and that trait can seem to spread through a whole population of bacteria. This horizontal gene transfer was something that fascinated New York scientist Joshua Lederberg, just starting out his research career in the late 1940s, when studies on genetics had reached a dizzying frenzy. The structure of DNA had not yet been published, but by the 1940s genetics was in full swing, and an increasingly large body of evidence had shown that genes were made of DNA. But how exactly this all worked was still largely unknown. If you were a scientist interested in DNA, there was an absolute banquet of questions to ask and mysteries to unravel. What Joshua Lederberg found was that this horizontal gene transfer was possible because the bacteria physically transferred DNA between each other. They get close together, and a special structure extends from one cell to the other, and a piece of DNA is transferred. A relatively small piece, just enough DNA to house a few genes. The bacterial cell has a single large chromosome that in a typical cell is about 5 million DNA bases long, enough to encode about 4,000 genes. In comparison, your genome, and mine, is about 3 billion bases long, encoding about 20,000 genes. But anyway, the bacterial chromosome is not transferred, nor any of the genes on it. Instead, the thing that is transferred is not a piece of the chromosome that breaks away. It is a separate entity that remains separate from the chromosome, like a little sister chromosome that floats next to it in the cell and can be transferred easily to other bacterial cells. Lederberg published extensively on this work, and in a paper in 1952, in the journal Physiological Reviews, he proposed a name: plasmids. This was not seen as an obscure piece of knowledge at the time. It was heralded as important work to unlock the secrets of life, so much so that he won the 1958 Nobel Prize in Physiology and Medicine. But still, neither Lederberg nor anyone on the Nobel Prize Committee foresaw just how big of an impact this discovery would cause. In 1967, researchers at UC San Diego used electron microscopy to actually see a plasmid for the first time. Electron microscopy uses electrons to bypass the inherent limitations of light microscopy. Light waves are each about 500 nanometers apart from each other. That makes light microscopy not really useful for seeing anything smaller than that size. But since a bacterial cell itself is only about 1000 nanometers long, so twice the size of the wavelength of light, light microscopes do not let you see all the little bits and pieces and details inside the cell. Electrons shot in a beam are only about 0.1 nanometers apart from each other. Basically, that means that using electrons you can see things that are about 1,000 times smaller than you can with light. Obviously, our eyes are only set up to see light, not electrons. But if you get a machine to capture the electrons and turn that into a picture, then basically, yeah, you can see with electrons. And the machines that do this, electron microscopes, were invented in the 1930s and have become accessible in many research labs by the 1960s. And plasmids, viewed under an electron microscope, well, they look like little circles. And I'll link those first 1967 pictures in the show notes. Little circles of DNA that can move between bacteria. And at the same time as this, in 1967, another scientist from New York, Hamilton O. Smith, was working at Johns Hopkins School of Medicine trying to understand and describe a special type of enzymes. Remember, enzymes are proteins that carry out biochemical reactions. He was interested in enzymes that cut DNA. Over the 1960s, several research groups had shown that bacteria make enzymes that are able to destroy the DNA of invading viruses. Yes, bacteria get infected by viruses. Other researchers had found that there are enzymes that seem to destroy all DNA. But what Hamilton O. Smith was able to show was that his enzymes cut only specific DNA. He used the very simple and rudimentary techniques available at the time to actually sequence the DNA that was being cut. I say simple and rudimentary just by comparison to what is available today. Today, if I have a piece of DNA that I'm interested in, I can pop it in a plastic tube, have it picked up by one of several local companies, and by the next day get a computer file with the complete sequence, the order of hundreds A's, G's, C's, and T's of my DNA piece. Instead, it took Hamilton O. Smith and his colleagues about a year to work out the sequence of just about 6 letters, DNA letters I mean, that formed the target site of the particular enzyme they were studying. They called the enzyme HIND3. There is a logic to that name, but I'll spare you the details. They found that HIND3 cuts any DNA molecule where it finds the following sequence of DNA bases in this order: AAGCTT. And once this was published, other researchers found more and more of these enzymes, each with its own particular target DNA site. They are called restriction enzymes because they were first discovered because they restrict the ability of viruses to infect bacteria. Remember this because in about 5 episodes' time, we're going to meet CRISPR, another genetic engineering tool that also naturally is part of the bacterial defense system against viruses. So by the mid-1970s, scientists knew about little circles of DNA, plasmids, that could be passed in and out of bacteria. They knew about special enzymes called restriction enzymes that cut DNA in specific places. When I say scientists, I don't mean all scientists. Long gone were the days of scientists having a good knowledge of every field of natural science. By the 1970s, following a trend that just continues to strengthen today, scientists have to become narrowly specialized in subfields of subfields, just to cope with the huge volume of specialist knowledge and specialist equipment and techniques necessary for their work. The genre of scientists I'm talking about are molecular biologists. Molecular biology is biology that focuses on the major biomolecules of genetics, DNA, RNA, and proteins, as the key material to ask and answer questions. By the 1970s, molecular biology had grown to a very prominent position within life science, and today I would go so far as to say that most biologists are molecular biologists of some form or another. But molecular biology is now so all-encompassing as a way of doing biology that it is barely useful at all anymore as a way of describing a specific group of scientists and their methods. But in the 1970s, it was still just one of many branches of biology, alongside the classical geneticists who still focus on the math of inheritance, the cell biologists who study whole cells, their components and how they work together, the anatomists who study larger bodies and how their bodies work, and the ecologists who study the many organisms and their interactions and a systems approach, and many more fields besides. And all of these other non-molecular biology fields still exist, but molecular biology is the largest partly because of the incredible work in the 1970s that showed how powerful molecular biology can be. It is powerful as a way to understand living things, and it is powerful as a way to reshape life itself. There was just one more ingredient needed to make it possible to reshape life with molecular biology. Some sort of molecular glue that could stick together pieces of DNA cut up with restriction enzymes. That comes in the form of another kind of enzyme called DNA ligase. And wouldn't you know it, that was discovered in the late 1960s. It was all coming together. And the person who happened to be first to put all these pieces together was Paul Berg, introducing the cold open. In 1972, he published cutting-edge work bringing together all of these pieces. He showed that you can take two distinct pieces of DNA from two different viruses, cut them out of their original viral genomes using restriction enzymes, and combine them together using DNA ligase to create a complete plasmid, or circle of DNA. And as you listen to that, it might instantly become clear why people would have been alarmed by this finding. There's nothing inherently scary about cutting up DNA and gluing it together. But what could you do with it? Could you create a supervirus? If you mentally fast forward to 2020 and the idea that the SARS-CoV-2 virus was made in a lab, whether or not that is true, it is not impossible. It's exactly that sort of dangerous and reckless action that the folks at the 1975 Asilomar conference wanted to make sure would not happen. The new viral DNA plasmid that Berg made wasn't a supervirus, it was totally harmless, but it was still a scary idea. The next shocking technological development was just a year later and led by a researcher working down the hall from Paul Berg, Stanley Cohen at Stanford University, and he was collaborating with Herbert Boyer at UC San Francisco. What they showed is that you can create a plasmid from different DNA pieces just like Paul Berg, but they also showed that you can take that plasmid and you can deliver it into a bacterial cell where that DNA is then understood by the cell's machinery and used for gene expression.
Cohen and Boyer
took plasmids from two different bacteria, cut them into pieces, recombine them together into a new plasmid with pieces from both originals, and then successfully put this recombinant plasmid back into one of the species and showed that it was successfully replicated.
Cohen and Boyer
both saw that there was potential to this technology, perhaps to create useful products like medicines. And their institutions, UCSF and Stanford, did not hesitate to file a joint patent on the technology. For a few years, neither Cohen nor Boyer knew what to do with their invention. It just wasn't the done thing for a professor in 1973 to pursue a commercial enterprise while remaining in academia. But luckily enough for Boyer's financial future, a few years down the line, a venture capitalist with a particular interest in molecular biology hunted him down and took him out for a drink at a San Francisco tavern. Bob Swanson, the venture capitalist, saw the potential and he pushed Boyer to see it too. And together they founded Genentech, the world's first biotechnology company in 1976. Amazing, except that they didn't have any products. Sticking together pieces of existing bacterial plasmids was a neat trick, but it wasn't by itself particularly useful. Raipur is a small village within a vast agricultural region of what is today Pakistan. In 1930, it was part of British India, and Ganpat Rai Khurana, the local agricultural tax officer, was insistent that his son, Har Gobind, go to the school and focus on his education. And so he did just that, studying hard, first in Punjab, and then later pursuing a PhD in organic chemistry at the University of Liverpool in the UK. He was ambitious, hard-working, and intelligent, moving between several research groups in Europe before securing his own research lab at the University of British Columbia in 1952. He worked on a number of problems, applying his organic chemistry skills to questions of molecular biology. But most relevant to this particular episode is his work in DNA synthesis. He painstakingly developed methods to build DNA building blocks from chemical parts and build together the chains of nucleotides that form a DNA molecule. In 1968, he showed the Nobel Prize for Physiology and Medicine, but he did not let that slow him down. In 1970, he published the first complete, totally lab-based synthesis of a yeast gene, 77 nucleotides long. Just like with DNA sequencing, the synthesis of DNA is today a very ordinary thing. I could order a set of 100 different DNA molecules, each 1,000 nucleotides long, and have them shipped to me within a week. In 1970, Khurana's success was rightfully seen as a monumental achievement. Because of this relatively new possibility of synthesizing short DNA segments, Boyer and Swanson had an opportunity. It was ambitious, but just about possible. They wanted a gene that encodes something very valuable to a lot of people who might pay good money for it. And then that also would be heralded as a great achievement for humankind, something that would make them look like heroes and make them rich. The answer was actually pretty obvious: human insulin. About 4 in every 1,000 people have a fatal condition in which they cannot produce insulin, which is the hormone necessary to trigger the processing and storage of excess sugar in the blood. Since the 1920s, the condition became survivable, with the option of injections of insulin harvested from the pancreases of pigs or cows. But this was not at all ideal. It wasn't well received by the patient's body, as well as being expensive and unreliable. In addition, by the 1970s, an increasing number of Americans suffered from type 2 diabetes, where the body loses the ability to produce insulin over time, and insulin shots in this case can also be life-saving. Insulin is a protein-based hormone. At this point, in 1974, no one actually knew the DNA sequence of the gene that encodes human insulin, but they knew that the protein is a chain of 51 amino acids. Since the genetic code 3 DNA bases are needed to encode each amino acid, that means 153 DNA bases, plus another 3 to encode the stop signal to finish creating the DNA chain. Boyer and Swanson knew that their best option was synthesizing the gene, even though they didn't quite know how. Swanson hunted high and low to assemble a team of scientists who could undertake this work. They first published, but did not try to commercialize, a proof of principle with the much smaller protein hormone somatostatin, that is only 14 amino acids long, so about a quarter of the length of insulin. But they showed that they could insert synthesized DNA encoding somatostatin into a plasmid, transfer that into bacteria, and have the bacteria produce the somatostatin protein. That work was published in 1977. And just 1 year later, Genentech scientists have shown that they could pull off the same trick with insulin. Unfortunately, to get the money to fund all this research, Swanson had pre-sold the rights to this product to pharmaceutical giant Eli Lilly. So it was Eli Lilly who began selling recombinant insulin in 1982, something they continue to do to this day. Genentech didn't slow down. They continued their work and created their own products, like recombinant human growth hormone in 1985. Today, Genentech still exists, but as a subsidiary of another pharmaceutical giant, Roche, which it was acquired by in 2009. But this podcast isn't all about biopharmaceuticals. It's about genetically modified crops. But the two stories are inextricably linked. By this time, in the mid-1970s, plant breeding was a huge business. Farmers across America and much of the world relied on companies to develop new improved crop varieties using the latest cutting-edge genetic science. And these companies were always happy to integrate the latest tools, such as the use of gamma radiation to create new DNA variation, as shown in our last episode. And so, of course, the idea immediately struck people that this new recombinant DNA technology could be part of crop breeding. And indeed, not too far away from all the action in San Francisco, over in Davis, California, Ray Valentine had a research lab where he was busily trying to apply recombinant DNA to agriculture in an approach he called molecular farming. Born in the Great Depression in a sharecropping family in Illinois, Valentine was a true believer in industrial agriculture as a force for good. He was also the only crop scientist invited to the 1975 Asilomar Conference, so it probably isn't too surprising that he created a company that released the world's first GMO crop. We'll pick up that story in episode 11. In our next episode, we'll dive into some of the additional science and technology beyond just cutting and gluing DNA that it takes to genetically engineer a plant. So we'll leave off here in the early 1980s with a world transformed. Over the 1970s, molecular biologists have shown that genes found in bacteria, found in humans, plants, and all other things are fundamentally the same. You can take these genes, you can move them between species, and they work the same. There's no secret sauce. DNA can yield to human manipulation as long as you have the right tools. Over the previous centuries, humans have become experts at taking chemicals out of the earth and turning them into everything from fuel to pigments to food supplements. But chemistry can only do so much. Over the 20th century, the geneticists have shown with increasing sophistication how you can guide the movement and recombination of DNA down generations through selective breeding. And that could already do a lot to reshape the traits found in species. But now the possibilities had been dramatically expanded. No longer limited by the genes that already exist within each lineage, shaped by billions of years of evolution, now scientists could possibly take DNA from any species, and if you could get the technical details right, deliver it wherever you want. What to do with that technology, and what not to do, are questions we are still grappling with to this day. We'll leave it there for now with our story. This has been Modified by me, Dr. Orlando DeLange. If you're enjoying the podcast, please do subscribe and tell a friend. The number of listeners has been inching up, but if you know someone who you think might be interested, do let them know. And if you want to get in touch with me, please do. You can email me at orlando@modifiedpod.com, and you can find my Blue Sky and Instagram handles to message me that way over at the website for the podcast, modifiedpod.com. And now, to round out the episode, here are your 5 take-home messages. Number 1. Bacteria can naturally exchange limited numbers of genes on small mobile DNA molecules called plasmids. Number 2. Restriction enzymes are used to cut DNA molecules at defined positions based on a short target sequence. Number 3. DNA ligase is an enzyme that can stick together free DNA ends. Number 4. The combination of restriction enzymes and DNA ligase can be used to create recombinant plasmids that can be moved between cells, the foundations of recombinant DNA technology. Recombinant DNA technology, this is number 5, is not by itself dangerous, but it allows researchers to modify living cells in dramatic ways that could be dangerous if used unethically. And with that, I hope you'll listen again soon. Bye!